This presentation is called The Pandemic Pivot, Teaching a Non-Major Science Course Through COVID-19. I'm using the word through in two contexts here. The first is that I was teaching through the time of the pandemic. The second is that I was teaching through the lens of COVID as a civic issue. I will discuss my experiences teaching a course called Molecules of Life. This is a 2-semester course for non-science majors at New York University. The course examines the intersection of chemistry, biology, and health with a focus on biological molecules. My first class of the spring semester was January the 28th. Started the class with a news story about a novel type of virus that had recently been detected. At that point, I had no idea that this virus would have such a profound impact on me, my students, and the course. Just before spring break, NYU announced that it was closing due to COVID. There was a frantic scramble as students left the university, and we made an abrupt switch to remote teaching. This map shows where students were located for remote instruction. Most students were within the United States, but some had to return to their home countries. After leaving the United States, many international students had to enter a 2-week quarantine in their home country. This photograph shows a quarantine cot for one of my students who returned home to Vietnam. During this time, many international students struggled with the challenges of remote instruction. I was living and teaching in New York, the epicenter of the coronavirus pandemic. As March progressed, the number of COVID cases kept increasing and hospitals were overwhelmed by the sick and dying. My class presentations on Zoom were often punctuated by the loud siren of an ambulance passing outside my window. How should science educators respond to such a difficult moment? I thought about how the Sensor Project had emerged over 20 years ago in response to another pandemic, HIV/AIDS. So I modified my course syllabus to include a 2-week module on viruses at the end of the semester. We began the virus module with a documentary about the 1918 influenza pandemic. After watching this documentary, students were asked to write about the similarities and differences between the 1918 influenza and our current experiences with COVID-19. The scientific content of the virus module began with 2 familiar examples: rhinovirus, which causes the common cold, and influenza virus, which is responsible for the flu. We also examined HIV as a case study for developing antiviral drugs based on the knowledge of the viral life cycle. As a civic issue, we investigated activism and advocacy by the LGBTQ+ community. This shows a demonstration by ACT UP to protest the high cost of AZT, the first AIDS drug, and the issue of the cost of COVID treatments and vaccines will certainly be a challenge for our current moment. The virus module then transitioned to an examination of the novel coronavirus, SARS coronavirus 2. This topic was challenging to teach because information was coming so fast, both from media reports and scientific publications. In fact, the World Health Organization called it an infodemic of information overload. This is an example of using the pedagogical framework of Sensor to teach through COVID-19 2 foundational scientific principles. Remdesivir is an antiviral drug, and that allows us to investigate principles of viral replication, enzyme function, and the structure of RNA. We also developed an authentic bioinformatics lab project for the virus module. Students compared the genome sequence of the 2003 SARS coronavirus with the sequence of the 2019 coronavirus. They also analyzed the genomes of different novel coronavirus strains to track geographic spread and evolution. I used the Sensor SOG assessment instrument to gather information about students' knowledge, skills, and attitudes, administered a baseline SOLG survey before we began the virus module, and a second SOLG survey 2 weeks later at the end of the module. The baseline survey asked students what they wanted to know about the novel coronavirus and COVID-19. The responses are shown here as a word cloud. Students were interested in how the virus spreads and how people are infected. I used the SOLG instrument to administer a pre-post assessment of students' understanding about viruses. There was a large self-reported increase in understanding for 9 areas of scientific content. 2 examples are shown here: how viruses infect cells and how antiviral drugs work. Based on the SOLG survey, students reported an increase in their ability to understand news media reports about the coronavirus and COVID-19. They also reported an increase in their ability to use reliable sources to find information about these topics. One of the challenges of teaching non-major science courses is that students often don't see the relevance of studying science. However, the SOLG survey demonstrated that, as a result of the virus module, students reported large gains in their ability to connect their general education courses to their major and their ability to apply what they learn in their classes to life outside the classroom. As the last question on the final SOLG survey, students were asked about the most important insight that they gained from the virus module. The responses are shown as a word cloud. Many students commented on the value of scientific thinking and the importance of being properly informed during the time of COVID. In conclusion, the educational ideals of Sensor provided an engaging and effective pedagogical framework for teaching students about viruses through COVID-19. Thank you for listening to my presentation. I hope you found it more interesting than my cat, who slept through the whole thing.